Good morning, everyone. Welcome to Breakfast Club number 9. So I think there are probably just 2 really good ways to start Mondays when times are this weird. One is by heavily spiking your coffee, which of course we would never do. The second is with stories of science trying to solve big problems, and that will definitely do. And joining us today is Dr. Rebecca Albright, Curator of Invertebrate Zoology at the Academy, and expert in how coral reefs cope with change, and co-leader of our Hope for Reefs initiative. Hey, Rebecca.
Hi, Laurel. Hi, everybody.
Thanks for joining us. Before I hand it over, I think you grew up in Ohio, is that right?
Correct.
Okay, so we all know Ohio is flanked by exotic oceans and diverse coral reefs. So how did you actually find your way to marine science?
You know, I think it's funny. I get asked that all the time, and I actually think it was growing up in Ohio that probably led to that. There's a lot of listeners that flock to the coast. And so I think it was just this kind of fascination with the unknown that always drew me out of the Midwest and towards the coast.
Oh, that's cool. Well, hopefully we can snag another kid with this today. Okay, so a reminder to everybody watching, whether you're on YouTube or Facebook, you can ask questions for Rebecca at any point just by leaving them in the comment section. And when she's done, we'll circle back and ask as many as we can. Get them in quick this time because we have a very hard stop at 11 o'clock Pacific because Rebecca's got an important task that she'll tell you all about. And with that, I'll turn it over for Hope for Reefs, creative solutions for addressing the coral reef crisis from restoration to cryopreservation. Thanks again, Rebecca. We'll see you in a bit.
Thank you, Laurel. Hi, everybody. Thanks for joining me this morning. And I hope that this presentation finds you all safe and well amidst these crazy times. I thought that what would be nice to talk about this morning is not necessarily just the dire situation that reefs are in right now, because I know that we don't really need any more doom and gloom amongst us at the moment, but to talk about some of the solutions that are being applied and creative things that are being thought about in terms of trying to help corals through these next couple of decades. So what I think we'll do is just start kind of with an overview, really basic stuff about why reefs are important, why we actually care about saving these ecosystems, and then talk about what stressors they are actually facing right now very briefly, again, not kind of dwelling on that doom and gloom too much, and then giving an overview of a lot of the different creative ideas that are being explored right now by researchers all over the world to try to help buy corals some time. And then I want to end with focusing on where the Academy is really trying to contribute to these efforts. So thanks for joining again, and I hope that you find this interesting. So most of the time when we think about corals, we think about something like this. They're one of the most iconic ecosystems on the planet. They're just teeming with life. They're incredibly beautiful, and most of us really value them for their aesthetics. We love to go diving, we love to vacation on them, but it's worth mentioning all of the different ecosystem services that they actually provide us every day free of charge. So coral reefs actually support about 30% of biodiversity in the ocean. And that's a disproportionate to the amount of area that they actually cover. They cover less than 1% of the ocean floor, but they're supporting 30% of biodiversity. So really punching above their weight class there. They mitigate wave energy for us, which actually provides us with human security and coastal protection. So it's been estimated that as little as 5 square meters of intact coral reef can mitigate up to 97% of wave energy. And so if you have a cyclone or a hurricane that's building energy as it moves across the ocean floor, it slams into that coral reef before hitting the coastline, mitigating all of that energy and storm surge, and actually protecting coastal infrastructure and human lives. They are the primary food source or protein source for hundreds of millions of people around the world, supporting fisheries. And they also provide us with tourism revenue. And if you wrap all of these different ecosystem services up, it's almost $400 billion of annual goods and services that coral reefs offer us, in addition to just their incredible beauty and wonder. So it's really something that we're worth protecting. And so a lot of the techniques that we're going to talk about today actually rely on manipulating different parts of a coral, either the animal or the algae that live inside the coral or the bacteria that live on top. And so I thought we'd just take a second to really break it down and talk about what a coral is, so that some of these techniques make a little bit more sense. So a coral is actually an animal, but it is also a symbiotic relationship with an algae that lives inside of the animal. So this is a close-up of a coral colony, and every little one of those tiny little white dots, I don't know if you can see my cursor, but every one of these little white dots is an individual coral polyp. And corals are related. They're in a group of animals known as Cnidaria. They're related to jellyfish and sea anemones. So it looks like a tiny little sea anemone. And here you see hundreds to thousands of these little polyps all living in association with one another in a clonal organism that we collectively call a coral colony. They communicate with one another. They share energy. They share resources. They reproduce in combination with one another. And if you break it down and look at just one tiny polyp out of this colonial organism, it might look something like this schematic here. So this one is, by example, about 2 millimeters in width, but different coral species have different size polyps. And this actually has a mouth, it has a gut, it has tentacles that ring the outside of the mouth. And so it can actually, and those are actively moving, it can grab animals and zooplankton out of the water column and then pull them in and ingest them. And then inside of the tissue of that animal lives an algae. And this is a single-celled dinoflagellate. In the upper right there's a picture of several cells of these dinoflagellates. And that algae lives inside of the tissue of the coral animal and provides it with food. So it's this fascinating symbiotic relationship that's developed over evolutionary scales, time scales, where the algae photosynthesizes and creates sugars, carbon sugars, that then translocate to the host, the coral. And the coral can get up to, in some cases, 90% of its energy needs from those algae. And this is important when we talk about coral bleaching later. And then in return, the animal, through metabolism, excretes wastes in the form of nitrogen and phosphorus, and the algae use those nitrogen and phosphorus sources for growth. So it's this very tightly regulated nutrient cycling and carbon cycling that happens inside the animals to provide them both with benefit. And it's an obligatory relationship. If this relationship is not functioning for most coral species, the animal will die. So something that's always fascinated me about corals, I think that they are the most amazing animals on the planet. And one thing that I just find mind-blowing still after almost 20 years of studying these animals is that they start life as tiny little larvae that resemble sesame seeds. So this was originally a video, but it didn't convert, unfortunately. And imagine these little sesame seeds swimming around in this water droplet. And each one of those is going to form one tiny little coral polyp. And then over time, this is a coral polyp that's recently settled. Over time, it'll start to grow a skeleton, and it'll start to bud asexually. It'll bud new mouths. So this is one coral polyp that originally settled from one larva that started to bud 1, 2, 3, 4, 5 new mouths. And through asexual budding, that'll continue to grow up into a larger coral baby, up into eventually a coral colony that has hundreds to thousands of polyps. And then you have hundreds to thousands of coral colonies that form a reef. And then you have structures as large as the Great Barrier Reef that are 2,300 kilometers long and you can see from outer space. And the fact that these structures start from little tiny larvae the size of sesame seeds and through geologic time form these massive structures that can be seen from outer space is something that continues to just be a wonder to me. And these incredible systems are at risk. So they're experiencing unprecedented rates of decline. Unfortunately, you may have seen this kind of plastered all over the news over the last 5 to 10 years or so, and it's picking up pace with global warming and climate change. And so it's been estimated that we've lost about 50% of coral reefs, and if we're on track to lose about 90% over the next 30 years if we don't do anything. There's a variety of reasons that cause this. There's local stressors like development. So this is actually an airstrip for a landing strip for airplanes off the coast of an island built right on top of a coral reef there. So development, overfishing and extraction, and this is both local subsistence fishing and large scale fisheries that put pressure on reefs, and then pollution and trash. nutrients and runoff, pollution, trash, all of those things cause stress on reefs. And then kind of the elephant in the room right now for coral reefs is global warming and its fallout, which is coral bleaching. So we're just going to spend a minute to talk about that. And so right now, CO2 levels, carbon dioxide levels in the atmosphere are higher than they've ever been in human history. And so this is the first time in human history that that CO2 levels are encroaching on 415 parts per million in the atmosphere. And this is showing the trajectory since the late 50s of CO2 in the atmosphere at a long run site in Hawaii that measures CO2 on a daily basis. And what's CO2 is a greenhouse gas, so it traps heat in the atmosphere, and about 93% of the heat that would be in the atmosphere is taken up and absorbed by the oceans. And so the oceans do an incredible amount of thermal regulation for us in terms of controlling climate because water has such a high heat capacity. So over 70% of the planet is covered with water. And so 93% of warming has actually been absorbed by the oceans. So that's an incredible service to us in terms of regulating climate, or else the atmosphere would be much, much warmer. But it's been estimated that the oceans, as a result of this, have already increased by about 1 degree Celsius over the last 100 plus years. And we're on track for them to increase over 2 degrees Celsius total by the end of this century. And that doesn't come without consequences. So unfortunately, we've seen more and more scenarios like this, where you have on the left a previously healthy coral reef that then over the course of weeks to months became a completely bleached and white reef. Here are a couple more images. This one was taken by one of our teams of divers from the Cal Academy. I believe this was in Micronesia. Luis Rocha, who gave a talk about a week ago on the Breakfast series, either took this photo or is the diver in this photo. And this was over the course of just a couple of months that this reef bleached. And so what is bleaching? Bleaching is just a stress response. It is a stress response that can be experienced in response to a variety of different stressors. It can be cold water, it can be too much light, it can be salinity stress, it can be dark. You can dark bleach corals, but right now what is causing widespread global mass bleaching is warming temperatures. And what happens, we think, during a bleaching response is the algae that inhabit those corals' tissues become a little bit overreactive, either because of light or because of temperature or a combination of both. And they start producing what are called reactive oxygen species. And the coral can't metabolize these or handle the magnitude of these that are being produced by the algae. And so what the coral tries to do, much like what you would try to do if you had a pathogen inside your body, is get rid of it, purge it. You try to get rid of it as quickly as possible. So the coral animal actually just expels the algae into the water column. And the algae, which are brown, give the coral much of its color. And so when it expels that algae into the water column, it looks completely white, because what you're seeing underneath is the calcium carbonate skeleton underneath that transparent animal tissue. So the coral's not dead. At this point, bleaching is not death necessarily. It can recover if the stress is taken away within a certain period of time, but it just lost its primary food source. And so if it doesn't recover those algae within a certain time window, it will effectively starve to death. And so we have experienced 3 global mass bleaching events. Like I said before, bleaching happens all the time in patchy little local isolated incidences. There can be a stress, you'll have a coral bleach. It kind of happens all the time, over and over and over. What doesn't happen in global mass bleaching events, where large surface areas or large portions of reefs all over the world are bleaching at the same time. That shouldn't happen. And that only happens in response to a global stressor that's affecting everything simultaneously, like global warming. So we've experienced 3 of these events so far. The first one was in 1998. The second one was in 2010. And the third one started in 2014, and we thought it would just last a year, and it just kept going, and it lasted through 2017. This is an example of what the heat map looked like throughout the world during that event, over 70% of reefs worldwide were affected by that third global mass bleaching event. So this is what's causing concern right now. Not just that corals are bleaching, but that they're bleaching all over the world simultaneously with the frequency that they're bleaching right now. And right now, as we're listening to this presentation, the Great Barrier Reef is bleaching for the third time in 5 years. And that frequency of bleaching events should not be happening. So they're actively monitoring this situation. These are reports that were from CNN and the New York Times just about a week and a half ago about the Great Barrier Reef bleaching event again. And we're estimating that these bleaching events are occurring about 5 times more frequently than they did 30 to 40 years ago. And the problem here is that reefs are slow growing. Corals are slow growing animals. They calcify these skeletons. They take time to build actual reef structure. And it's estimated that after a bleaching event, it takes about 10 to 15 years for a reef to recover. And so when you have a frequency of bleaching, like 3 times on the Great Barrier Reef every 5 years, you're just not getting a window of recovery that's long enough to have any sort of meaningful progress in a recovery direction. Okay, last bit of bad news, and then we move on to solutions. So this was a report that was released last fall by the IPCC, the Intergovernmental Panel on Climate Change. It was a special report organized by the United Nations, and it was looking at the effects on different ecosystems if we were to cap global warming at 1.5 degrees Celsius. And so remember, we've already increased by about 1 degree Celsius since 1880 or the pre-industrial era. So if we can cap it at 1.5 degrees, what do we risk losing? And then if we actually go towards 2 degrees, what do we risk losing? And what they found through this series of models and assessments was that coral reefs are one of the most vulnerable ecosystems. Arctic sea ice is also incredibly vulnerable. And if we're able to cap it at 1.5 degrees, which is very unlikely at this scenario, but if we're able to do that, we're risk losing 70 to 90% of coral reefs worldwide. And if we creep towards 2 degrees Celsius, then we're actually looking at effectively losing everything, over 99% of coral reefs. And so here's where we are right now, is facing these kind of realities and recognizing that right now, the only real solution to this at a global scale is mitigating climate change and cutting carbon emissions as quickly as possible. That said, we have to do something else to try to buy us time. And so we have to do something to try to support coral reefs through these difficult times so that once we are able to gain traction at meaningful scales with climate change and cutting emissions, that there's more to recover from. So that's what we're trying to do. And so we all know the saying, necessity is the mother of invention. Here it is from another great philosopher, Calvin and Hobbes, waiting for last minute panic to strike creativity. But, the thing that gives me the most hope in situations like this, for a few years there, it was actually really hard to be optimistic about all of this. And there has been palpable changes in the scientific community over the last 5 years that, I've been doing this about 20 years. And the change that has occurred in the way people are thinking and the types of things that people are trying to do over the last 5 or so years is incredibly inspiring. And we haven't necessarily found a solution yet, but I think that the type of thinking that's happening is on the right track and is really, really inspiring. So that's what we're going to focus on for the rest of this talk. I want to give you again an overview of all of the crazy ideas that are being talked about. And then we're going to focus on where the academy is kind of keying in. So a couple of examples of really great initiatives that never happened before. Just a couple of years ago, we had the first ever International Coral Restoration and Intervention Science Symposium. So there's a new kind of consortium called the Coral Restoration Consortium that just formulated a couple of years ago. I'm part of this consortium, and we hosted the first ever international conference to try to talk about solutions. This was in Key Largo, and it brought hundreds of coral scientists from all around the world to get together and talk about how are we going to solve this problem together. And that was something that hadn't really happened before. Similarly, the National Academy of Sciences processed a consensus study report looking at all of the different interventions and ways to increase persistence and resilience of coral reefs. And some of these we'll talk a little bit about today, but it included everything from more conservative approaches like reef restoration techniques and managed breeding techniques, all the way up to some pretty alternative and futuristic scenarios like phage therapy or microbiome manipulation, or actually mixing cool water onto reefs in order to try to have local mitigation of warming. So all of these different things are being explored from conventions of reef scientists all the way up to the National Academy of Sciences. And actually, just about a week and a half ago, the Australian government rolled out this new plan called the Reef Restoration and Adaptation Program, the RRAP. And this pledges $150 million to support, I think it's the 43 ideas that may promise to provide resilience or support resilience and adaptation in coral reefs. And again, these kind of break down into three different buckets. One is shading reefs and cooling reefs. One is supporting recovery and adaptation on reefs. And one is actually facilitating adaptation a little bit more proactively. A lot of different things in this bucket as well, from things that are, again, a little bit more futuristic, like cloud brightening, which is spraying microparticles, nanoparticles of salt into the air in order to try to get more light scattering so that you don't have as much light penetration onto reefs, which promotes local or regional cooling and lower incident light levels to mitigate bleaching. And then some more conservative things like just using concrete blocks in order to stabilize structure on reefs that's been compromised from either wave activity or cyclone damage or bleaching events. And that was just last week that was rolled out. So to talk about a few of these things, one of the things that you might have heard about is something called supercorals. And so a question that gets asked quite a lot is, well, there are corals that live in more tolerant environments. There are corals that live in the Red Sea that regularly reaches 35, 36, 37 degrees Celsius. Can't we just take all of those corals from the Red Sea, breed them like crazy in coral farms, and then outplant on them all over the world? And boom, we have heat-resistant reefs. It's not quite that simple, but we'll talk through some of these scenarios of things that people are trying to do. So again, just reminding you that the coral is actually what we call a holobiont. So there's the animal itself, the animal tissue, the coral animal. There's the algae that lives inside of it. And then there's the bacteria and microbiome that coats it. And so all of these different parts of the holobiont are things that people are looking at manipulating to try to build resistance in coral colonies. So one idea that has been proposed is breeding to create hybrids of corals that are more tolerant to stress. So this is an example of using two different Acropora species and creating a hybrid. And there's a concept called hybrid vigor, where the hybrid or the F1 of two different species that were crossed is actually more resilient to stress than either of the original parent species by itself. And so this is one example, is to try to hybridize coral species to create new species that are more stress tolerant. Another example is manipulating the microbiome. And so moving corals from cooler to warmer environments and seeing whether there are shifts in the bacterial community that the coral hosts that might promote thermal tolerance. I have a student right now that is doing a reef restoration project in the Dominican Republic, where she outplants corals to different environments and looks at the shift in the microbiome of those corals to try to understand how much that microbiome is actually regulated by the host and how much it's regulated by the environment. And if that's something that we can manipulate, like give corals probiotics to try to infer different physiological traits that we want. Another thing that's being proposed is adapting the algae instead of the corals. So as we mentioned before, corals are really slow-growing animals. Usually corals, kind of the more fast-growing species, like a branching coral that's here, like an Acropora or a staghorn or elkhorn coral, will grow maybe 8 to 10 centimeters a year, best case scenario. And a mounding or like a brain coral will grow maybe 2 to 3 centimeters a year. They're incredibly slow-growing. And reproductive cycles are incredibly so. So they usually only sexually reproduce once a year for most coral species, and it can take about 4 years in a best case scenario to get from a baby coral up to a coral that's actually reproducing itself. So that you can imagine, if you're interested in adaptation and evolution, that's a long time. When you have bleaching on the Great Barrier Reef three times in 5 years, you don't want to wait 4 years to get another generation of corals that might be a little bit more resistant. The algae that inhabit the inside the tissue has a much shorter generation time. And so those you can actually keep alive in the lab, expose them to different temperatures, try to select for populations of the algae that are a little bit more heat tolerant, selectively breed those, and then perhaps inoculate corals back with those heat tolerant strains and try to infer thermal resistance in the coral itself. So that's something that's being explored right now. Some of you may have heard of CRISPR, which is genome editing, whether you can actually introduce portions of genes into a genome to actually create some sort of bleaching tolerant coral. And so they're not actually trying to create bleaching tolerant corals using CRISPR right now, but CRISPR has been done in corals. As of a couple of years ago, a group at Stanford led the charge on this, and is actually able to do genome editing in corals. And so whether or not that'll play a role in the future is going to be interesting to see. Cryopreservation is another technique. So this is something that's been developed for a lot of different animals that have conservation breeding programs, as we need to know how to cryopreserve germplasm and cells. And so this is something that Mary Hagedorn from the Smithsonian has been leading the charge on for the last 5 or 10 years. And the Coral Restoration Consortium that I mentioned just a few slides ago has actually just formed, they have different working groups of scientists to try to lead the charge on different areas that we deem are critical to the future of coral reefs. So for example, there's a larval propagation working group that I serve on for the Coral Restoration Consortium. And they just started a cryopreservation working group, or it's in the process of being started as of the last couple of weeks. And I'm going to serve on that one as well. And the Academy is going to try to pitch in to that effort as well. And this is to really develop techniques to learn how to cryopreserve coral material. So Mary's been very successful in learning how to cryopreserve coral sperm. So there's about 800 coral species, we think, and she has been able to successfully cryopreserve about 16 of those species, so about 2% of the biodiversity of corals. We can successfully cryopreserve the sperm. The larvae, the eggs, and actual coral fragments are a little bit more challenging, but we want to be able to move towards that. And that's part of what this working group for the Coral Restoration Consortium is going to try to do over the next several years. And then here's just a little appetizer of a bunch of other things. So there's a Sunshield trial that this is developed by people in Australia that were involved in developing the Australian money, which is a plastic. So it's a polymer it's a biodegradable polymer that could be sprayed over the surface of seawater at large scales and kind of help with light reflection, so that you get less light penetration into the shallow water and help protect corals from coral bleaching. This was an effort that I was involved with. I led this work on the Great Barrier Reef to try to, this is dealing with ocean acidification, but we tried to reverse the effects of ocean acidification locally to try to enhance calcification and growth on coral reefs, and showed that we were able to do that. Whether or not it could be applied at scale is debatable. These are some larval bots that are in development by a group led by Pete Harrison, again in Australia. A lot of this work is coming out of Australia just because of the Great Barrier Reef. But the idea here is that these robots, much like a crop duster, these robots would go and collect larvae during spawning events, and then actually like little AUVs, autonomously operated vehicles, just to be deployed over the Great Barrier Reef or over areas of reef where you would want to release coral larvae in mass and have them settle. So it's a way of trying to spread larvae around. And I'm going to talk a little bit more about this in a second, but this is Dr. Taryn Foster, who is currently at the Academy working with us. And she is a Fulbright scholar who's working on using robotics and automation to try to scale reef restoration efforts, which is really exciting. And this paper I just wanted to point out was just released last week, I believe. And it's actually printing bionic corals. So this is really cool. What this group has done out of Cambridge has used CT scanning in panels D and E. So A is an actual coral, B and C are actual corals, the skeleton and the tissue. And then D and E are CT scans of the skeleton and the coral tissue, and then they used a 3D bioprinter to actually print bionic corals so that to mimic at a micron resolution the detailed architecture of the skeleton. And one of the potential applications of this is to host coral, the algae, the zooxanthellae that the coral needs inside of these bionic materials, and try to evolve them that way to understand bleaching mechanisms and whether there's anything that we can learn from in order to try to mitigate bleaching. So two of the processes that I have been the most interested in throughout the course of my career have been growth and reproduction. And this is mostly because when I started grad school, we were already experiencing unprecedented decline. So I started grad school back in 2005, and in 2003, there was this very iconic paper called Gardner et al. 2003 that was released that said that in the Caribbean, we lost 80% of coral cover. And that was kind of the first paper of that kind. There have been many since, but that was, as someone that grew up, like Laurel said, in Ohio, that just was fascinated with these ecosystems. And I didn't get into this to be a conservation scientist. I got into it because I love them. And then I entered grad school because I love corals and I wanted to study corals. And then right as I was entering grad school, all of this incredible decline started happening. And so because of the timing of all of that, I got really, really interested in recovery processes. And the recovery of any population is contingent on growth and reproduction. Those are the two things that have to happen in order for any species, any population to be self-sustainable. And so very quickly, I'm going to look at just introduce how coral reproduction happens. So corals reproduce asexually, which we saw a little bit at the beginning. They can bud and clone asexually within a colony. And then you can also break off a coral branch, and that branch will start to regenerate, much like a starfish arm. And then the original coral will start to regenerate. And then sexual reproduction happens two main ways, spawning and brooding, but we think about 75% of coral species reproduce via spawning, which I'm going to talk about in a moment. And so we've taken advantage of these reproductive modes for reef restoration efforts. So reef restoration has been around for about 30 years. Most reef restoration efforts happen using asexual fragmentation. So you see coral gardens or nurseries like this underwater, where you take certain corals, break off branches, repropagate them, and grow them in mass like that. And then you can replant those corals back onto the reef. One of the things that is worth mentioning with this type of approach is that you are creating more coral cover on the reef, which helps sustain those ecosystem services, but you are only propagating a single genome. Well, not a single, but you're propagating, you're continually propagating whatever genotypes you started with. So if it's not partnered with sexual reproduction, which is the way that we get new genetic diversity, then you have a limited amount of genetic diversity in these systems that, or these restoration efforts. And so partnering it with sexual reproduction is really quite important. This is called microfragmentation, which they discovered at Mote Marine Lab several years ago. And what this is really cool, again, that if you, instead of fragmenting larger chunks of corals, if you fragment centimeter-sized pieces, because of an inherent regeneration or repair process, we think the coral experiences something akin to a wound, and it goes into kind of an overdrive from an immune perspective. It will grow about 50 times quicker than a larger chunk that's broken off. And so what people do when they use this type of microfragmenting technique is break off tiny, tiny little pieces of coral of all the same genotype. and they grow 20 to 50 times faster than they would in the wild. And then because they are the same genotype, just like skin grafting, if you then put them close to one another later, they will fuse. So you can get coral colonies that are much larger than you would in the wild much more quickly using this technique, which is really cool. The other way that corals reproduce, so this is sexual reproduction, is called broadcast spawning in most coral species. So this is about 75% of coral species experience sexual reproduction this way. And once a year, they release gametes, so eggs and sperm, into the water column. And those gametes fertilize, form larvae that then eventually swim, settle onto the reef, metamorphose, and start to asexually bud and grow into coral colonies. And we're partnered with an organization called SECOR, which stands for Sexual Coral Reproduction, that actually tries to help corals through this process, because spawning in the wild is actually breaking down as coral populations decline. So you can imagine that you might have one coral colony spawn, you used to have densely packed corals all close to one another. And when they would spawn, all the gametes would be right next to one another. They would fertilize, they would mix, you'd get tons of coral larvae. Now, with how depauperate some coral populations are, you might have a coral colony here, another one over there. And if you do get synchronous spawning, they're too far apart sometimes to mix or to fertilize. And so this organization called SECOR is going out into the wild to help facilitate this annual reproductive event. And so what they do is go out and net coral colonies when we know that they're going to spawn. And when we spawn, the gamete bundles float up into these little collector cups, which we can take off and bring back into the lab. So what you see here are tubes filled with eggs at the top, and the sperm is in this cloudy water down below. There's an up close of some of the coral eggs. And then you can cross-fertilize them to create little coral larvae. Here's a large vat of embryos being fertilized. And then once the larvae are large enough, you can settle them. And then this is a little baby coral that looks like it's maybe 12 different mouths at this point that will eventually be planted back out onto the reef. So what this organization is trying to do, and the Academy of Sciences has partnered with them through our Hope for Reefs initiative, is help corals, shepherd corals through these vulnerable early life history stages to enhance post-settlement survivorship and the number of baby corals that we're getting back out onto the reef. So this is an example. This was a paper that was published a few years ago where some of those corals that were planted back out onto the reef by SECOR actually grew up and spawned with their native populations, which was so exciting. And here are some divers out planting corals back to the reef. So one of the limitations with these restoration techniques, and one of the problems that the Academy is helping to try to address, is scalability. So as you've seen from the beginning of this talk, the scale of reef degradation is global. The Great Barrier Reef is 2,300 kilometers long. And it's been estimated that about 50% of that reef has been impacted by these events, has died over the course of the last 5 years through these back-to-back bleaching events. So we're really good with reef restoration techniques of putting corals back out onto the reef, at restoring corals to healthy states at about a hectare scale. But you can imagine that this is ultimately dependent on an individual diver going and planting, like epoxying or cementing a coral onto the reef, and that's extremely time and labor intensive. And so we have to figure out ways to try to scale this, to try to get out of that hectare scale all the way up to ecologically meaningful scales. So one of the projects that we've been working with over the last 5 years has been a partnership between the Academy and SECOR and the Autodesk Foundation to try to come up with seed units. So these are units that we settle coral larvae onto that are self-stabilizing, so that we don't have to go physically epoxy them to the reef, but you could scatter them. And they are like jacks from the old game, but they're self-stabilizing, so they move less than about 50 centimeters over the course of 6 months. And we're looking at different designs for different species and different types of reefs, coming up with a bunch of different options to see what works best for different scenarios. And the idea is that you condition these in mass, so hundreds to thousands of these in the ocean, and then you settle larvae onto them, and then you would go out and you could actually either throw them out over the side of a boat and they would sink down and self-stabilize and then not move very far, or you could deploy them underwater via diving. But either way, it's removing that step of actually having to physically fix it to the reef surface. And here are some baby corals on the left, and then a little bit of larger one that's actually adhered itself to the reef through this project. So that's one really exciting project that the Academy is involved in. And we have sites in throughout the Caribbean, and they've actually expanded into Palau and the Indo-Pacific with this collaboration. Another really exciting effort that we're doing at the Academy is supporting Dr. Taryn Foster from Australia. So I mentioned before, she's a Fulbright scholar, and she is here for a year working with us and Autodesk in the Autodesk residency program to try to use robotics and automation to scale reef restoration. And so this is using the asexual methods of fragmenting corals would be the idea, and using that microfragmentation technique where you can get reskinning, where you put smaller pieces of coral that are the same genotype next to one another, and then they all refuse. And so Taryn's idea is pre-printing the majority of the skeleton for the coral. So what we talked about before is that a coral colony through asexual budding might take 4 years to get from a single polyp all the way up to a colony that's large enough to then reproduce. And Taryn's basically hacking this process to say, you know what, I'm going to give it the pre-made skeleton. So this is a brain coral example here, or a dome-shaped morphology of a coral. I'm going to give it that skeleton, and then we're going to put coral fragments using the microfragmenting or another fragmenting technique and plug them into this pre-made skeleton. And then it'll graft over the surface in a much shorter period of time than building the entire thing itself. And then you get a coral that can be outplanted to the reef. And the idea is that these prototypes have been developed with Autodesk and with the intent that they are robotics and automation friendly. So you would be having these plugs inserted by a robot, and then the actual base of the frame can be manipulated by a robot. And that's, again, removing some of the human labor aspects. So really cool stuff there. So stay tuned to that. Her project is called Coral Maker. And the last thing that I want to talk about is a project that I've been working on for the last couple of years, 2 to 3 years at the Academy, called the Coral Spawning Lab. And this is kind of been my baby since I've been there. But the goal here is to try to get corals spawning in a conservation breeding, try to develop conservation breeding techniques to spawn corals in a land-based setting, to try to get that annual reproductive event happening in a land-based setting. And that's something that may sound simple, but it's not, and I'll show you why. And it's something that has only been done in one other place in the world so far. So what we're doing is, again, developing these protocols for predictable broadcast coral spawning in a land-based setting. And so what we have to do is mimic all of the different environmental cues that elicit this one time a year reproductive event. And so it's generally thought that there's a lot of different things that we think play into triggering this reproductive event, but it's generally accepted that there's 3 big ones that cue into when corals are going to spawn. So corals usually spawn in late summer, after a full moon, a few days after a full moon, a few hours after sunset. And that's staggered depending on the species, but that's the general recipe. And so what we need to mimic is seasonal changes in temperature, because we have to have the corals experience a winter and a summer. The actual process of oogenesis, which is creating eggs inside of the coral, can take up to 7 to 9 months in some species. And we think that it's that warming in late summer that triggers the maturation of those eggs in order to get them ready for the spawning event. So they have to experience those changes in temperature over the whole course of the year. Then they have to experience full moons and new moons. And so because most coral species spawn a few days after a full moon. And then they have to experience sunrises and sunsets because most coral species spawn a few hours after sunset. And so these are all things that we're trying to mimic in an aquarium setting. All of these different things that are on this slide are things that we think influence reproduction. And so we're trying to simulate a lot of these things in the Steinhardt Aquarium inside the Academy's walls. So the 4 that we have been focused on the last couple of years are seasonal changes in temperature, lunar cycles, day length, and solar insolation. So we use this programming system called, it's by Neptune, it's called Apex, and they've so generously donated most of their equipment because they were so excited about this project. And we sourced our corals from Palau. And so what we did was for Palau, we have 10 years of historical data from NOAA saying what the average temperature, seawater temperature is every month of the year. And we program the average month seawater temperature for January, February, March, all throughout the year into the system. And then every day, the system is iteratively changing just a tiny little bit to create the next month's average temperature. So those temperatures are mimicking the seasonal cycles. Then we program in the day of the new moon every single month and the times of moonrise and moonset. Again, this is all for Palau because these corals were collected in Palau. That's mimicking our lunar cycles. And then we're mimicking sunrises and sunsets and day length by programming in the exact time of sunrise, the exact time of sunset. And we have LEDs. We have lights that have 8 different wavelengths of light, 8 different channels. And we can change which channels are experiencing which intensities at different times of day. So for example, at sunrise, you might want a little bit more red light. At peak noontime irradiance, you might want more white light. And so we're changing those parameters to try to get them to experience a sunrise. And we ramp that up over the course of 3 hours. So 180 minutes, then they experience more white light and full peak time, noontime irradiance, then a sunset, and then moonrise and moonset all throughout the year. And then we're also changing solar insolation. So the actual percentage or amount of light that they're getting throughout the year based on what the tilt of the Earth would be relative to the sun and solar insolation. So all of these things are being manipulated in our system. I'm going to show you this short video that a student made in my lab about the process over the last couple of years. This is just a couple of minutes, and then I have a few final slides before we end for today. This is the process of making that. So because we are mimicking these light settings, we have to have complete control over light pollution. We had to build a room within a room. It's like an old photography dark room so that we don't get light pollution from anywhere else. Then we sent a team of divers to Palau to collect coral colonies. So we have 4 different species in the spawning lab. We're mostly working with a species called Acropora hyacinthus, and we always collect them sustainably. So we never take an entire genotype off the reef. We target large colonies and take no more than 1/3 of that colony off the reef so that we're leaving. And again, remember, because of regeneration, both sides of that should regenerate. They're shipped overnight to the Academy of Sciences. They're in transit about 30 hours. And then we put them into this dark room where we're mimicking all of those things and try to simulate the conditions that they need in order to spawn. And so this is a picture or a video of mass spawning that happened in our system last year. So we've had spawning in our system the last couple of years. This is last year's spawn. Those are the gamete bundles being released from the corals. And then this is us doing what I showed you that sea core does. So we're fertilizing the eggs and the sperm in order to create embryos and create coral larvae, which we then settle to create coral babies. Oy. This is early stages of development. The embryos become ciliated and they start to spin, and then they elongate and they'll start to actually swim. This is a very delicate and vulnerable process that we try to shepherd them through. These are the little baby coral larvae swimming around and settling. choosing a place to set up shop for the next X number of years. Hundreds of little baby corals settled onto a puck that's probably about an inch and a quarter in diameter. And then they start to take up that symbiotic algae within the first few weeks of life. And I just want to share with you some results that we've had from last year and this year since this video was created. So so this picture I took on Saturday or Friday, just a few days ago, and these are 1-year-old baby corals that are the product of last year's spawn inside the sign heart. And so these are Acropa hyacinthus, and just for size reference, those discs that they were settled on are about 1.5 inches in diameter. And so these are a year old at this point, which is really exciting. And we're really hoping that eventually we'll get them to that 3 to 4 year stage and that they'll actually spawn with their parent colonies as well. This is, so the way that we check to see whether or not the corals that we collect from the wild are gravid, which basically means that they're going to reproduce, is we break a branch off and you can actually see the eggs inside of the coral colony. So this is a branch that has been broken off of one of the corals that we shipped over from Palau this year. And these are the eggs that you see with those arrows inside of that coral colony. So this is what we're checking for. And so the last few years, what we've been able to do is we're shipping these coral fragments over from Palau. We keep them in our system for about 2 months. And then, but we verify that they're gravid before we ship them over. We keep them in the system for about 2 months, and then they spawn at the same time as the wild parents in our system. And that's really exciting. But obviously the end game here is not to take corals off the reef, but to actually get these coral colonies conducting the entire reproductive cycle inside the academy's walls. And so what we really want is for them to develop eggs over the course of the whole year by themselves. And this is something that I just saw the last couple of days, that we're actually able to do this, which is, I'm just over the moon with this. I'm so excited. So this is a coral colony that was collected last year. It spawned last year and created those 1-year-old corals that you just saw. And this is a branch that I took of it on Saturday. And those are eggs inside. So this means that we are actually able to elicit the entire oogenic cycle. And I'm right now going in every single day to monitor for spawning from these corals to see whether the corals collected this year and last year spawn at the same time. So what do we do with it once we actually get spawn? This type of system can cater to so much research, which is really, really exciting. But some of the things that we're actively doing right now are contributing to the optimizing land-based conservation breeding protocols. So these are techniques that were developed. I mentioned that this has been done one other place in the world, at the Horniman Museum in London. So we're using a lot their techniques mostly, and we're helping try to optimize these techniques because this is for most conservation breeding programs, understanding how to keep animals viable year-round in a land-based setting is really important from a conservation standpoint. We're also going to be using this system to help develop cryopreservation techniques with the Coral Restoration Consortium working group that I mentioned before. And we're looking at techniques that can be used to help optimize and maximize growth and survivorship of baby corals so that we can translate that knowledge into the Sea Corps work that's done in the field to try to get better survivorship and growth of baby corals and field-based restoration. It also offers, because we have access to all the different life stages, you can do so much just from a basic research standpoint too, that then will eventually provide tools for more applied research. So we're developing a genome for this coral species. We are looking at germline segregation. We're looking at how the microbiome changes during reproductive events. We're looking at sperm activation and calcium ion channels that are involved in activation and how it relates to other other animals over the evolutionary tree of life, all sorts of different things. And these are just projects that we're doing this year, because I'm going in during COVID every day to check on these. And the idea eventually is that you could have multiple systems like this and stagger them to get reproduction multiple times a year, and enhance the pace of scientific discovery. So I want to end there because we do need to stop in a few minutes because I have to go in and monitor the corals for coral spawning. But I wanted to let you know that the Academy has been amazing in supporting a coral spawning camera that you can see live if you type in the URL at the top of this screen. And so they're on a reverse day night cycle. So during our day, it's their night, and during our night, it's their day. So if you sign in from 9 p.m. to 9 a.m., it'll look like on the left, and you'll actually be able to see the corals and the fish. And if you sign in from anytime from 9 a.m. to 9 p.m. during our day, it'll either be completely black. Apologies for that, but we don't want to mess up the circadian rhythm and photosensory mechanisms of the corals. Or I turn on red lights when I'm in there checking. And so I'm going to rush out of here in about 20 minutes and go in and check. And so if you were to sign on to this webcam within the next couple of hours, intermittently, you'll see red lights come on, and you'll see me in there with a little red light looking to see whether there's any spawning activity happening. So anyways, thank you so much for joining us this morning. I hope that this has, first of all, distracted you and maybe provided a little bit of interest during this difficult time for everybody. I know we're all struggling a little bit at home, but also I hope this provided you with a little bit of hope in terms of the outlook moving forward for the future of coral reefs. So thank you very much, and I'm happy to answer any questions you might have for the next 5 or 10 minutes.
Rebecca, thank you so much. And for anyone watching, we're going to go ahead and drop the hyperlink for the cam that Rebecca referenced into the comments section to make it easy to click. And if you want to bookmark that in case you see some action in the next few days, please do. So I'll ask, let me try to get in a handful of questions. We won't get to all of them, but we can kind of circle back in comments later to try to answer the ones we don't get to. Sarah G asked, do we know what cues the larvae use to decide where to settle?
Yeah, that's a great question, Sarah. So there are a bunch of different cues we think. We don't, but the one that we think is most important is that there's an encrusting algae, lots of different species, but generally it's called crustose coralline algae, that certain species of that corals really seem to like. And it's not just corals, larvae of a lot of different marine invertebrates seem to want some sort of chemical compound that's in that algae, or it might be the bacterial that's on it, and in order to settle. And so we culture that as well, actually, in our lab in order to facilitate that settlement event.
Okay, cool. Let's see. So Julie asked, and this is specific to the super coral, if you use them to replace a reef that had died, would all the original fish come and other things the first reef supported come back, or would it become an entirely different ecosystem?
That's a great question, and it's actually something that we don't know. And so we do know that fish communities generally are catered to whatever food they have in that direct environment. There are some fish that are coralivorous, which means they eat corals. And so it would depend on what type of corals you put out, would probably dictate the fish community that was part of the fish community that was there, not all of it. But those kind of unknowns are part of the reason why it's not permitted right now to release supercorals into the environment, or there are a lot of repercussions that we don't fully understand. But those are things that are being considered as we talk about actually implementing some of these at scale.
Okay. So Andrew's question, I think what he's basically trying to understand is why reefs, if it's getting too hot for them, sort of why they aren't just starting to grow in places that are traditionally a bit colder on their own.
That's a great question too. So a couple of different points to that. One is corals have limited ability to move. So they are fixed to the ocean floor. They're not like fish. They can't move around. The only time that they actually can move around is in the larval phase. And again, that usually only happens for most corals once a year. And we don't really, there's some evidence that some coral larvae will stay in the water for a very, very long time. But there's also evidence that some are very local. So limited ability to move. But the other thing is that warming is only one of many stressors. And so warming is obviously worse right now around the tropics, but as you get towards higher latitudes, ocean acidification becomes more of a problem because colder water hold more CO2.
Mm.
And so there was a paper published a few years ago that showed that this latitudinal range of like where corals would be able to live is narrowing because you have temperature pressure from the equator northward and southward, and then you also have ocean acidification coming up from this side that's making it more corrosive and more difficult for corals to survive.
Wow.
So I think that helps answer that a little bit.
Okay, it does, and it also addresses a question that Stephen asked along the same lines. Jay Gomez asks how does dive tourism affect reefs? And he says he always wanted to visit them, but felt conflicted about disturbing them. Can it be done responsibly?
It can be done responsibly. And so there are, Belize actually has a really great tourism program where you have to, so you pay as part of your tourism fee, you pay a fee when you go diving that goes back into supporting the marine park, and that's obligatory. You have to pay that as part of your dive ticket. And you have to, in some countries, demonstrate a buoyancy test before they'll let you even on the reef. So most of the pressure from diving comes from people maybe kicking the reef or, inadvertently injuring the reef. And then there's, just boat pressures and things like that. But there are places that are doing it a lot better than others. Usually there's a fee to support the marine park and then some demonstrated competency of divers before you're unleashed onto the reef.
Okay, those are two good things to look for. Great. And I'll just hit you with two more questions and then we will let you go because you've got other babies to tend. Laura D asks, so she's a marine biology master student studying coral, and she asks, what skills do you think would be best to focus on? Genetics, stable isotope tracing, anything particular you'd recommend that is important or in demand?
That's hard.
Are you checking the webcam between questions? You're just like...
No. I think it would be remiss of me to say that if I didn't say that right now, kind of big data is like we're collecting data at scales that we don't have the ability to process. And so skills related to molecular science and processing all of that data, like bioinformatics skills, are really, really valuable to be able to just understand all the amount of information that we're getting. And that was something that when I was in grad school, 15 years ago, it wasn't really on anybody's radar so much. But now it seems like you can't really get very far without having some sort of molecular and bioinformatics skills available. So I would say that's probably, there's a lot, Laura, right? I don't know. But
Laura.
I would say that's one thing I can't imagine that's going to become less important.
Okay, great. Yeah, I'm sure that'll be helpful. Okay, this is a big question, but it seemed like a good one to end with. AJ asked basically just what the average person can do to help.
That's a great question. And I had a few slides on that. Actually, I can hear. So, a few things that we always say at the Academy that you can do to help that just really seem tangible, that actually make differences are, one is be an informed consumer. So if you consume things related to the oceans like seafood, there's this amazing seafood app called Seafood Watch in Monterey Bay. So definitely use that. Make sure that you're consuming things that are sustainable and aren't putting additional pressure onto coral reefs. That's one very easy thing to implement. There's an app. And then this is something that is in our planetarium show called Expedition Reef. That is an example of your impact based on your food choices, which is easy to implement every single day. You have at least three, however many times you eat a day, options to make good choices that might ease pressure on reefs in terms of carbon emissions. And so this was a little schematic that our planetarium came up with to show you how far you would have to drive if you made a different food choice. If you ate a plant-based burger, like a veggie burger, I think it's a turkey burger, and then a beef-based burger. And so this is San Francisco, if you're not part of us, but this is Golden Gate Park right there where the Academy is. And if you chose for lunch a plant-based burger, you would just be driving, a few blocks in terms of the amount of carbon that was released to produce that burger. If you chose a turkey burger, you'd be driving to South San Francisco area, and then a beef burger would be kind of further down the peninsula, and then you scale it up to making that choice every single year, these are the equivalent amount of carbon emissions. So point being that nobody's perfect. You're not going to do this all the time, but being an informed and aware, conscientious consumer, and then as often as you can, making choices that are more sustainable and minimize your impact on the environment. There's 7.7 billion of us, and if we all do that as frequently as possible, it would ease the pressure on not just coral reefs, but on ecosystems worldwide.
Great. Okay, thank you. Yeah, we'll wrap it up there. That the mention of Expedition Reef reminds me, I think we actually have that online. So if we do, I'll drop a link to that in this as well, so people can watch that show at home. But yeah, thank you so much for joining us. I know you have to run off. Everybody keep an eye on that Coral Cam and on our social accounts for updates. And Rebecca, thank you again.
Yeah, very happy to be here. Thanks so much. Be well.
Yeah, for sure. And viewers, we have coming up this week on Wednesday, we have a Earth Day special where we're going to talk about the largest citizen science movement in the world. And then on Thursday, we're talking herpetology with Dr. Raina Bell, who is a frog expert, and going to just dive into herpetology 101. So it should be a good week. But this is all about corals, and we'll keep the updates coming throughout the week. And again, Rebecca, thank you.